Hello guys, hope that you're good. Um, Today I'm going to discuss about different receptors that are present on the surface of macrophages and help with different functions of the macrophages. But before that, just to talk about the receptors that are present on cells of the innate immune system, there are more than 100 receptors that are present on the surface of these cells of the innate immune system, including macrophages, natural killer cells, dendritic cells. Not all of the 100 receptors would be present on a single cell type, but on different cell types, there would be multiple receptors present, multiple sets of receptors present on them. So, what is the function of these receptors present on the cells of the innate immune system? They are going to distinguish between carbohydrates or lipids or nucleic acids that are part of the microbes and not part of the mammalian cells. Okay, so in other words, they are telling cell from non-cells that what are the carbohydrates, which carbohydrates or lipids or nucleic acids are part of the mammalian cells and which are not part of the mammalian cells, and they are foreign bodies. So if they are foreign bodies, the body or the innate immune system cells of the innate immune systems are going to act against that particular foreign body or entity. And this discrimination leads to more precise induced response against the microbes on infected cells. I have told you about the innate immune responses, that there are two types. One are the immediate innate immune responses, which would take 0 to 4 hours, and they are carried out with the help of preformed molecules and effector cells. The preformed molecules, which are already prepared there for the purpose of countering different types of microbes, or for instance, the complement factors which are there, which bombard the pathogens or which label the pathogens to be phagocytosed by different phagocytes like the macrophages and neutrophils, etc. So the other type of innate immune responses are the induced immune responses, when the immediate innate immune response is not enough to neutralize or kill the pathogen, then the induced responses would be initiated. And these macrophages are very important in that respect as well. So then the induced responses would generate many different types of molecules that would actually um, invite other cells to the site of infections to help with the macrophages and clear that particular infection or that pathogen, to kill that pathogen. Okay, so these receptors, if you look at the number of receptors, there are just 100 receptors, but these receptors are responsible for recognition of pathogens or microbes that are in hundreds or thousands of different microbes can be recognized with the help of these 100 receptors. So just 100 receptors recognizing thousands and thousands of pathogens or disease-causing microorganisms. What is the trick here, and how are these receptors going to recognize such a large number of microbes? Um, that is because of the fact that these receptors are not specialized kind of receptors that would just go for recognition of a single peptide or a single, carbohydrate moiety. They are going to recognize a pattern of carbohydrates, a pattern of nucleic acids, a pattern of lipids, which are part of the membranes or walls of these different types of microbes. And these patterns would be common to them. For instance, a carbohydrate pattern, which is common to gram-negative bacteria, or carbohydrates found on the surface of gram-positive bacteria. So all the gram-positive bacteria would have that pattern, or pathogen-associated molecular pattern, as we call it. And the pattern recognition receptors are going to recognize all the gram-positive bacteria with that pattern recognition receptors that are going to recognize that pattern. And similarly, uh, you know, uh, uh, you know, other nucleic acids or lipids, these sometimes would be common to many different types of pathogens. They would be present in the membranes of many different types of pathogens. So it would be just like a generalized recognition by these cells. So they can effectively recognize self from the non-self, that this does not belong to the body. So that kind of recognition is enough at this stage to mount a response against that foreign body or that antigen, which the cells of the innate immune systems are very actively doing. So what is the trick? The trick is that they recognize the patterns on the surface of the pathogens with the help of these receptors. The receptors are fewer in number, and the PAMs, they are numerous, but these PAMs are generalized patterns recognized by these pathogens. So in uh, if you look at these receptors on the macrophages, there are many different types of receptors on the macrophages for recognition of carbohydrates, lipids, and proteins. There are scavenger receptors to clear the debris or the dead cells from the body, or the dead microbes from your tissues. And there are receptors for signaling of infections and to lead to the process of inflammation or to start the process of inflammation. And there are receptors for antibodies, there are receptors for complements. All these receptors are present on the surface of macrophages. So uh, if you look at the exact nature of these receptors, which are involved in different functions, for instance, the macrophages, they carry a battery of phagocytic and signaling receptors in all tissues. You know that they are resident macrophages. Macrophages are made, uh, in our bodies in the bone marrow, and then they are present in the blood as monocytes. We call them monocytes, and the monocytes, when they travel to the side of, to different tissues, then they would undergo many changes, and they would uh, you know uh, be turned into tissue macrophages. So there are resident macrophages in the tissues, and all the tissues have them, and they are permanent guards which are standing there and looking for any foreign body or any antigens that would enter the body. So any antigen or a microbe that would enter, that would cross the barriers of the innate immune system, like crossing the skin barrier, entering into the connective tissue, or crossing the mucosal barrier and entering into the connective tissue, would come in contact with these resident macrophages. And these macrophages are not going to attack these or recognize and attack these pathogens. These macrophages can also recognize and attack the commensals. The commensals are normal flora, normal bacteria, and other microbes associated with our skin and our mucosa, our mucosal membranes. They are numerous. The microbiota, about the microbiota associated with your gastrointestinal tract or your skin. So normally, those microbes are associated with our mucosa in the skin and are performing different functions, giving us different benefits and getting some benefits out of this relationship as well. But if these common cells somehow breach the mucosal membrane or the skin barrier and enter the body's connective tissue, then these commensals would also be taken up as antigens by the macrophages, and they would act against them with the same impunity. So the receptors that are going to help with these macrophages to attack these pathogens, to recognize or attack these pathogens are given here. For instance, these are the names of the receptors present on the surface of macrophages, and this is the process of phagocytosis that these receptors actually would help with, and how they are going to help with it. For instance, this is mannose receptor, which is also known as CD206, and this receptor is going to bind to certain substances present on the surface of bacteria, certain bacteria. For instance, the lipopolysaccharides or CPs or myelin, etc. These are part of the bacterial membranes, and these receptors, when bind the LPS, etc., this binding can lead to increased phagocytosis of that particular bacteria. Similar is the case with complement receptors. They are also helping with opsonization or phagocytosis of bacteria and fungi, because the complement receptors not only bind with complements but also with other oligosaccharides, proteins, and beta glucans, which are part of the bacterial fungal membranes. The dectin receptor, which actually is a carbohydrate receptor, also can bind, these uh, on the surface of bacteria and fungi. They can bind different carbohydrate moieties and help with phagocytosis or internalization of these microbes. Similar is the case with these scavenger receptors, scavenger receptor A or B, or the lipopolysaccharide receptors. All these receptors are helping with the process of phagocytosis, because these are the PAMs, or pathogen associated molecular pattern, right here under the name ligand. These ligands are all PAMs found on the surfaces of these bacteria, or these are either bacterial nucleic acids or bacterial carbohydrates or lipids, etc., that are going to be recognized by these receptors in the same. So many different types of bacteria or fungi which are sharing these uh, carbohydrates or lipids would be recognized the same way by macrophages with the help of these receptors. And there are signaling receptors which are known as the toll-like receptors. It's a family of 10 receptors with viable specificity for a range of pathogens. And these toll-like receptors are going to lead to initiation of this process of inflammation, which is a protective response of your body, in which the macrophage is going to signal other cells via chemotactic factors to come to the site of infection and fight off the pathogen or kill the pathogen. So a little bit detail about these receptors. The receptors on macrophages for the recognition of different carbohydrate moieties are known as lectins. Okay. Usually the receptors for recognition of the carbohydrates moieties are present on the surface of cells, and these receptors are known as lectins. And lectins are carbohydrate recognition receptors present on the surface of macrophages, are called the mannose receptor and dectin-1, these two types of receptors. So these two types of receptors, which can also be called as lectins of the macrophages, they have a carbohydrate binding domain, and that domain is a specialized type of domain where when the carbohydrate moiety binds, it needs the help of calcium ions. Okay, so the calcium, it means that this process of binding of the carbohydrate, which is present on the surface of the pathogen, with this receptor, mannose receptor or the dectin receptor, is dependent on calcium. So that's why we call it the C-type lectins. Okay, and the binding domain of the carbohydrate with the receptor is known as the C-type lectin domain, or the CTDL, because of the calcium. Okay, so remember the calcium here. So The different C-type lectins, they are C-type, receptors. They differ in the number of these C-type lectin domains, the binding domains which require calcium for binding. So, for instance, in the case of mannose receptors, there are 8 CTDLs, while in the case of dectin, there is only one CTDL. In mannose receptor, not only this is the domain for binding carbohydrate, there is another carbohydrate domain that binds sulfated galactosamine residues called, the R-type lectin. There is another domain, R-type lectin. This is the C-type lectin. There's an R-type lectinase. And why it is called R-type lectin in case of mannose receptor? Because this domain is structurally similar with resin, which is a highly toxic lectin or carbohydrate of the castor bean. Okay, so that's why this mannose receptor also has another domain for binding with carbohydrates, uh, and that domain is it is similar to resin is called R-type lectin, apart from the CTDL. There is another group of receptors on macrophages, and this group of receptor is known as the scavenger receptors. Why are they called the scavenger receptors? Because for the first time when these receptors were observed they were observed to scavenge LDL proteins from the blood, or low density lipoproteins from the blood. That's why they are called scavenger receptors. And these receptors recognize negatively charged microbial ligands, uh, which are different molecules that are negatively charged and that are part of the microbes. Okay, and there are two types of these scavenger receptors as part of the membrane of these macrophages, which are known as scavenger receptor type A and scavenger receptor type B. Uh, the type A, they are particularly recognizing LPS, or the lipopolysaccharide in gram-negative bacteria, teichoic acid of gram-positive bacteria, and the cytosine guanosine-rich bacterial DNA. These are the PAMs recognized by SRA, and these are the PAMs recognized by SRB, are the scavenger receptor type B. So if you look at these PAMs would be common to many gram-negative, many gram-positive bacteria, and many different types of gram-negative and gram-positive bacterial when it comes to their nucleic acids, for instance. Uh, there are other types of receptors present on the surface of macrophages, which are known as the macrophage receptors with collagenous structure, are Marco, that binds both gram-positive and gram-negative bacteria. For instance, this is Marco. I will just, in a slideshow show you this in a while, that how does this receptor look like. And if you look at the length of this Marco receptor, uh, you know, the Marco receptor looks, the stature of this receptor looks taller as compared to the complement receptor here, or the dectin receptor here in yellow, or the SRB receptor here. And this receptor in stature is closer to the mannose receptor, which has got two domains here, the CTDL and the RTLD. So one of the function of these receptors, just like the scavenger receptor A, uh, is to keep the pathogen at a distance from the surface of the macrophage, which would help, with uh, with good recognition of that particular, pathogen, so that it is recognized in a better way. So it keeps it away from the membrane. The domains here It has got a triple helix-like structure here. In SRA and the Marco, both of them, they share the same structure. So it binds both the gram-negative and gram-positive bacteria. Similarly, you know already in our complements lecture, I had told you that CR1 and CR2, they bind C3b. And where is C3b? For instance, this is bacterium. This will be labeled with complement factor 3b. And for C3b, there are receptors on the surface of macrophages and other cells uh, and which binds with C3b. So if the disease bacterium is labeled with C3b, it would be readily taken up by the phagocytes. This is called opsonization. CR3 and CR4 receptors also are complement receptor 3 and complement receptor 4 for the complement IC3b, which is just a fraction of the bigger 3b fragment. You also have gone through this process of making up the IC3b. So IC3b does not help with opsonization of the pathogens, but it can help in other ways, uh, these CR3 and CR4 receptors are going to bind lipopolysaccharides or lipophosphogycans in leishmania or hemagglutinin protein in Bordetella pertussis or glucans and candida. So many different types of pathogens can be opsonized with the help of these complement CR3 and CR4 receptors. These are important receptors here because all these receptors that you see here that are present on the surface of macrophages are somehow going to help with internalization of the pathogen. This is the mannose receptor here, and it has this R-type domain and the C-type domain. Carbohydrate that binds here would need calcium, and ricin-like carbohydrates would bind here. Similarly, this is dectin-1, and I told you about dectin-1 that both of these are used for recognition of the carbohydrates or binding of the carbohydrates take place here. Carbohydrates which are part of the membranes of pathogens. Similarly, this is the complement receptors. You can take it for CR1, CR2, CR3, or CR4. There are these different complement receptors present here. And this is Macro. You see this triple helix structure here. It looks similar to SRA, and this is SRB. And other receptors, scavenger receptors. The bacterium taken up with the help of the complement receptor because it is labeled here, opsonized here. The yeast would be taken up with the help of dectin receptors because it would have these carbohydrate moieties on the surface of yeast, and this would be just internalized, and these pathogens, once internalized in the form of phagosome here, this phagosome would be fused with a lysosome. And you know about the composition of things in the lysosomes, that they have acid hydrolases, and these enzymes can readily degrade these pathogens into smaller pieces, and the body can get rid of that. So that was about the scavenger and other types of receptors for the recognition of different PAMPs on the surfaces of pathogens by the macrophages. And tomorrow I will discuss about signaling receptors and how the signaling would, these toll-like receptor would sense the presence of infection, and how the inflammatory process ensues for clearance of the pathogen. Thank you very much.